to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 16th episode of the Weekly Bioanalysis. My name is John Perkins. I am the Senior Scientific Advisor for all things related to LC-MS technologies. I've been with KCAS for 10 months, but I've been in the bioanalytical space for over 25 years. I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I'm the Senior Scientific Advisor at KCAS with a focus on ligand binding assays, biomarkers, and flow cytometry. I've been with KCAS for about 7.5 years now, and I've been in the bioanalytical space for close to 26 years.
Today's podcast, as always, is brought to you by KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. Dom and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. We're thrilled to have you listening again. As mentioned, this will be our 16th week recording the podcast, so if you're interested, the other episodes can be found at kcsbio.com. As a reminder, you can follow us on Twitter at @TheWeeklyBio and at KCSBio. As we mentioned before, we kind of started this whole thing because of social distancing getting in the way of normal communication. Even today, Dom and I are in completely different places. I'm still in upstate New York, Dom is in Kansas City, and our producer is in a different part of the Midwest. This weekly podcast gives the chance to connect with all of you, and we hope it's a chance for you to get to know us, and even more, it's a chance for you to get to know KCS and the services we provide. As always, a review of latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're always looking for new topics, and we'd be happy to discuss something that you want us to cover. Then we'll wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off, Dom is going to go over today's podcast topics a bit before we jump into news and resources section. Dom.
Yeah, we have an exciting podcast number 16 here. The main portion of our discussion will be around the challenges of bioanalysis in non-routine matrices. And what we mean by that is we kind of think of it as a soluble one and then more in the tissue area. And we'll touch on things like ocular matrices, urine, and then we'll dive into tissue. So, John, we got a jam-packed show here, and let's just jump right into the news.
So, I found this week there was an awful lot that we wanted to discuss, but we really had to spend some time whittling it down to topics that made more sense for us to focus on. Remdesivir has been in the news on a number of fronts, but really the most interesting is that researchers are concerned about lack of diversity within the ongoing remdesivir studies. We've seen that minorities are disproportionately affected by the COVID virus, but in the trials they are severely underrepresented.
Yeah, absolutely, John. There continues to be quite a bit of news around remdesivir, and not only the lack of diversity within the clinical trials, and I could throw some numbers out there. It looks like the original trial of 397 people done by Gilead had a total of maybe it looked like 11%. And then some other studies were more in the 20 percentile, and then even some hard numbers around an Alabama study, which showed that of the 27% population African American, only 43, but they accounted for 43% of the positivity. So really some numbers that still have to be discerned and kind of teased out. And, I don't know. I think, COVID continues to be elusive around best way to treat it and, how it should all work.
Yeah, and just the effect on different populations, and we know from the trials we've supported, you really want to look at if a drug's aimed at a particular population, you want to know that it's safe for that population in hand, and if it is disproportionately affecting minority groups, we do want to know that they can deal with the therapies that are developed and approved. It's interesting because this sort of coincided with Bristol Myers Squibb announced that in the wake of Black Lives Matter and just looking at the history of the country in terms of minority representation, they've announced that they want to focus on ensuring that their workforce and at the executive level are more represented by minorities. And actually, within the article I read, this topic of Hispanic and Black people being underrepresented in clinical trials was something that they see as well. They don't have a plan to address it, but really, we're in many drugs in development, we're developing them based on a white population only, or substantially many more white clinical trial subjects than from minorities.
Yeah, definitely something that has to be addressed, and I hope there's some movement to continue to improve and grow as a country, and most notably in diversifying us. So interesting stuff, and we won't even go down the other drugs that are being used for treatment of COVID. We're just going to move on to a really interesting
Yep.
study that ranked all 28 vaccines. I really found this to be quite fascinating. Is a big mixture of companies on here, and what we won't not necessarily have to say all of them, but let's do some highlights. And from for several weeks now, we've been talking about how Pfizer and BioNTech were in the lead, and then Moderna was sort of right behind them. And then actually, I thought AstraZeneca and the Oxford trial were maybe in the lead as well, but it turns out that according to this study, that's the third place one, and then the Sinopharma China one, which we've highlighted before as well. And then I think we should, I don't know, you probably saw the news. Putin gave his daughter a vaccine, and Russia claims they have the vaccine, and that ranks fifth on this list.
Yeah, I'm going to treat that with a pinch of salt, but I have to say a big shout out to Nicole DeFudis, who is an associate editor at Endpoints News, who's the person who's put this list together. So basically, talking about the 28 main players, ranking them as they are right now, and I know Endpoints News is to sort of continually update this as things change. It's certainly interesting when you look at the 28, that 26 and 28, well, 26 and 28 are Sanofi and Glaxo, and then Merck in the final sort of 28th spot, which doesn't mean a lot. It's really saying where they are in terms of development. But it's the where you've had the high profile vaccines have gone very quickly. Both GSK and Merck are talking. We want to get this right, so we're not going to rush this. We want to have something that will be effective and follow a more methodical approach, even though the timelines they're talking about are still very accelerated compared to standard vaccine development.
Oh yeah, absolutely. And GSK does pop up with a collaboration with Clover Biopharmaceutical, and that corrects the sixth place, I think, is it on the list here.
Sure.
Just
Yep.
one that surprised me. This is Kentucky Bioprocessing. I had not heard of them. They're actually cracking the top 10. We should mention the Zydos Cadelia. That's in India. They're ninth. And then I think I saw Duke on here mid pack around 15th. And then there's a bunch of German companies like CureVac, a couple German companies. BioNTech, of course, is another one. But then the People's Liberation Army Academy of Military Sciences China was 17th or 18th. That one kind of made me.
Yeah. Because of course you've had China take the similar approach to Russia and basically administer their vaccines to a captive population, and skipping the phase 3 clinical trials. We are actually studying safety, etc. So we'll see how this changes. I think this is something that we'll revisit on a regular basis, and it certainly is interesting. And then another article that we found this week was actually in Bloomberg, but talking about vaccine production, and their Bloomberg's take, and it really is a fascinating article in terms of the family background, etc. They're talking about the Serum Institute of India, and the CEO is a guy called Adar Poonawalla. He's a billionaire, and effectively has gambled on the need for vaccines. So he's moved into a realm of the pharma industry where the margins are tight, but because he is personally wealthy, he can afford to go into these looking to meet unmet needs where other companies wouldn't necessarily go in that direction. And so he's gradually expanded the vaccine production capabilities of the Serum Institute of India, and is in a good position for when when the need is there for production to actually step in and help. I will definitely be linking this article on my LinkedIn profile, because it's a very interesting piece of background, particularly when they talk about the family money, and the grandparents had a horse farm, and they saw people producing anti-venom and other antitoxin sera, and realised that they had horses on site that they had retired, and they could actually put those horses to work producing antivenoms, etc., for further sale. So really, really interesting.
Yeah, a lot of, well, I mean, just on the horse front, we are seeing more and more horse antibodies are just a good source. So on the LBA side, we got quite a few reagents that are coming from horses now. I mean, it looks like they're going to be able to produce like 1.5 billion doses. I mean, goodness gracious, that is I mean, just a wealth of, or just an astronomical number when you think of it annually. And even today, I think they're equipped to do about 500,000,000. But then, they're gonna they have a 50-acre campus, right? I mean,
Yep.
50-acre campus. I mean, good God, John, that's like, I think my school was like that big, right? I mean, Jiminy Cricket.
Yeah, and then of course they're already signed with Oxford and AstraZeneca to produce a billion doses of that, and are actually then talking to other candidates that are in development. So having seen the name for the first time, because I'd never heard of them, I think I'll definitely be watching to see how often they pop up in the news, and if they do deliver and become a big part of the COVID story.
Yeah, and I think and didn't read the article too much. I mean, I skimmed it. I read parts of it. What was fascinating was how far ahead of the curve he was. Right.
Yeah,
That's what
absolutely.
makes the read so interesting. Is that he just, he could have simply lived on the money that he had. I mean, like you said, he was a billionaire, but instead he kind of put his money to use and took some risks. And I think people probably thought he was crazy because in order to have a 50-acre campus, that started back in '01, I think. And so I think he kind of felt the need, very altruistic individual, and really a good read if you can find it on Endpoints about the Serum Institute of
Yep,
India.
yep. So it's actually Bloomberg is the magazine. So if you can find the article on Bloomberg, that's where to go. I picked it up from Endpoints, but it is actually a Bloomberg article. So moving on, Moderna is back in the news, having scored big with a $1.5 billion coronavirus vaccine order. It's interesting. We see these big numbers being batted around, and of course we haven't shown that any of these vaccine candidates are actually effective yet, because they're still in development. But obviously, everyone's trying to do what they can to get ahead of the curve.
Yeah, this is mind-boggling to me. I actually was just checking out Moderna's stock for a random reason last night with a friend, and so it seems to have, it didn't, upon this news, it didn't trigger anything, but it certainly has skyrocketed. And they've never had a vaccine cross the finish line. Um, mRNA type vaccines in and of themselves are challenging. I mean, Alnylam has been in that space; they're popping up in it. But I don't know. I think it's one that very powerful, very potent. Let's just hope it doesn't have some of the side effects that other mRNA vaccines have had, or has some more specificity to it
Yeah,
versus just attenuated thing.
yeah,
Those are
I
basic.
know there's been concerns with higher doses on this, that they have seen side effects. So hopefully those don't amount to too much.
Yeah, fascinating. Before we pivot off of COVID, there is one more article about healthcare workers lose out on COVID sick leave protection, says a federal watchdog. And I think I talked to you before about how my brother was in getting treatment and somebody refused to see a COVID nurse. These are the types of things that's really discouraging. I hope that isn't the path they're going down, because who in their right mind is going to want to lose their any sort of pay or protection if you're a treating COVID patient. So that one.
Yeah, yeah. From my perspective, healthcare workers are the principal group you really want to rally around to make sure that they're protected as much as possible,
Mhm.
seeing as they're in the forefront of dealing with this.
My sister's a nurse, as I mentioned, in Mass General. Things have really calmed down there, but still, I just you don't penalize somebody who's doing great things, right? Like so.
Exactly.
That hurt. And John, I do think we're going to take one more minute and talk about sports and COVID here in the U.S. A substantial amount of football has been canceled. I kind of have been thinking all along that we'd see no fall sports. I don't think we'll be watching any sports live anytime soon here in the U.S. It's still to be determined. Some schools, like the University of Nebraska, threatened to go play despite that, but then they're in the Big 12. The Big 12 said you do that and you're out, and now Nebraska's drawn away from that. I do know, like for example, in Kansas City here, they've they're wait and see on sports now. So my son's football games are currently off, meaning he's not going to do them, but they're taking a wait and see approach. And I do know that the city of Omaha, Nebraska, canceled all fall sports, and that
Wow.
was troubling to hear.
Yeah, so UK equally has some updates on the sport front. Scottish football season has actually kicked off, but of the nations in the UK, Scotland has probably been the one that's most tightly locked down. But recently, 8 Aberdeen players went out to a bar together after a game, so basically broke lockdown, and then 2 of those players have been diagnosed with COVID-19. So there was there was obviously a little bit of a ruffle about that, and then it was announced basically revealed that a Celtic player had done a similar thing where he'd actually, I think, flown to Spain, come back, didn't self isolate, and then played the day after coming back. The knock-on effect is that the Nicola Sturgeon, who's the first minister of Scotland, has put Scottish football on a yellow card. That if they break any more of these kind of rules, then she'll be looking to call off the season. In England, it's a slightly different point of view. Team I follow, Hereford, are in the I think sixth level of non-league football, where the crowds are actually pretty sparse. Their season has been put off. And they're looking to start around October 3rd. Where right now, in a typical season, they'd have started already. So they haven't actually even played any pre-season friendlies. But the concern in non-league football is actually the fans really are the lifeblood of the teams. Because Hereford are actually reasonably well followed, where we maybe get 2,000 for a home game. A lot of non-league clubs, you'll maybe see 100 to 200 or less than 100 fans per match, but they're still vital to keep the clubs going. I mean, they're all run on a shoestring. So there's actually a petition is circulating with let fans in to non-league games, because they desperately need any sort of income they can get. So I do sympathise with them to some extent, because I've been to games where you're not near anyone else, and people are citing, if you look at photos of beaches, which we talked about in other podcasts, where the beaches are absolutely crowded. Non-league games, you're never like that number of people. In fact, non-league games can be quite charming, where you change it, as the teams change at halftime, the fans change ends as well, because there's all the space to get the best viewpoint of your team as they're playing.
That's awesome. Good stuff there. But obviously, COVID will to be determined around sports. I think in the coming weeks we'll have to address that more. But it's going to be challenging this fall, and I would be surprised if you see any college sports in the fall here in the U.S. So, John, we're going to do one non-COVID item. I know we're kind of running up against it here a little bit, but I really have a passion for TREM2. So what they've discovered is this. It's been in the neurodegenerative space as a hot biomarker, and what they're finding out, it's been implicated in Alzheimer's, and it's a marker we have. We have a method for it. We've tested several studies using looking at TREM2 levels in CSF and plasma and things like that. But now the University School of Medicine in St. Louis has shown that it is potentially good for immuno-oncology. So they have an anti-TREM monoclonal antibody along with a PD-1 inhibitor, looking to see if it can combat immuno-oncology. So really cool stuff there. Let's not lose sight of some of the excellence that's going on in science outside of COVID. And actually, there were quite a few other things that intrigued me this week, but let's just stop on that one. And most notably, because if you have any TREM2 needs, you can call us here at KCAS.
There we go. Yes, you should always use it for biomarker work.
Yeah. So we're going to move along to our main topic this week, and it's something that we have a lot of passion for here at KCAS, and that's the challenges of bioanalysis and non-routine matrices. That's a good way to kick off agenda for, or the main portion for podcast number 16 we're up to. And John, I think when you came to us, you probably started to realize just how much expertise we had in tissues. And, we think of them as non-routine matrices, but we are in the animal health corridor. We got a 40-year history here, and we've been working in this space for quite a long time. And so why don't you let us know what you've learned about tissues and some of the challenges and how it's all working from your side of the fence?
Well, not having had any hands on, I still haven't learned too much about it. I am very impressed about how people are willing to take on assays in non-routine matrices. What were referred to as non-routine for the bulk of the bioanalytical work that we do, we're really looking at blood-based samples, whether it's blood, which I would also consider non-routine, plasma, or serum. When we start talking non-routine matrices, we'll be looking at things like urine, which has its own analytical challenges, tissues. There's also for ocular drugs, you start looking at things like tears, which is also interesting to work with. Other, bronchoalveolar lavage is another one. So we could pretty much split them into two camps. There's fluids. Biological fluids that are non-plasma, quite often it's lack of proteins was what makes them challenging to work with, and then there's tissues, which are another step up again.
Yeah, that's well said, John. You're absolutely right. There's that soluble side of, the solution-based ones, and they lack proteins, or at least they have some viscosity to them that is unique and makes it challenging. Things like CSF and ball and ocular fluid, they all have some viscous nature to them. And then there's two types of ocular fluid, right? Aqueous and humorous. And so humorous, excuse me. So yeah, and the challenges frequently there is the levels are somewhat low. You got to be very careful of um, sometimes there's signal to noise issues that make it somewhat challenging, and then sample volume, right? So in the biomarker space, even in the
Sure.
PK space,
Yep. Yeah,
that
I mean,
becomes even more
tier.
challenging to test so many things, right? Go ahead, John. You wanted to add something?
Yep. So yeah, I mean, how should
Okay.
should we start with the fluid side of things? I mean, I threw blood in there, so even though it's not common for us to do blood assays, but every so often there'll be drugs that are actually much more suited for measurement in whole blood rather than plasma, and the challenge
Yeah.
there becomes pipetting. It's how can you guarantee an accurate aliquot, and it really comes down to pipetting technique to ensure that you get things right with blood.
Yeah, and on the LBA side, whole blood on flow cytometry is quite common, but we almost never do whole blood. We would do a whole blood cell lysate. So we're still spinning that down, or the site will spin it down and drop it into a freezer. And then a lot of we have several, not so much biomarkers, they're more activity assays, pharmacodynamic assays that we use whole blood lysate. So makes it easy for the site to just, spin down and decant all the supernate and leave a little bit on there, drop it into a freezer, and then ship it along. And then we do a BCA assay, and we'll talk more about that certainly when we dive into tissues, but that's a total protein measurement, and we are cranking those out. We do them daily, right? So, I
Sure.
mean, different challenges, I think, on the whole blood, and it's definitely a unique matrix on your side of the fence as well as on the large molecule side.
Yeah. So, moving on to urine, for instance, in the LCMS side of things, that's one particularly where lack of proteins in the matrix can cause a real issue. So what we find is many drugs, or if we're doing method development in urine, almost our first step is to determine whether we have non-specific binding going on. And what we mean by that is basically we have the analyte in urine, and you want to non-specific binding is basically adsorption to surfaces out from the liquid. So if you have, say, urine in a polypropylene tube with your drug, you'll find with time that the level of the drug seems to be dropping, and that's because it's adsorbing to the side of the vial. It can be a tough thing to address. We tend to use surfactants, or to to help with that. But some of that becomes then the challenge for the clinic is how do you prepare? At what point do you add the surfactants? Because your urine sampling is in big vessels, then you want to transfer that to the tubes. And then we're testing, even if you have adsorption, can you actually add the surfactant late on to disrupt that adsorption to bring your analyte back? And we had an assay. This is going back several years. We had an assay that we worked on for many weeks, where it was a parent drug in its metabolite, and actually the parent absorbed. They both showed non-specific binding, but we couldn't find a single agent that would disrupt both. So if you're just looking for the parent, you'd use one agent, such as maybe it was tween, something like that, and then for the metabolite, use something else. But to try and actually develop an assay for both at once was near impossible because of non-specific binding.
Yeah, no, John, that's similar on the large molecule side. We definitely, we can kind of overcome some of that in urine just with using a low binding tube. The bigger concerns are a couple things. One is interference with capture and detection antibodies. I mean, I think that's always a concern, but urine has osmolarity, salt concentrations, and pH that are different than the standard diluents. So if you're looking for, and typically you're looking for a low level of something, or you don't want to, you can dilute. The challenge is it's typically a large volume, and then we have to do an MRD on that. So sometimes you can run it neat, but those are the challenges that come with it because they collect a lot of volume. And then I think regardless of the platform, collection of urine, at least in human clinical trials, we're going through one now. You got to worry about just contaminants, right? And I'm not going to go down what that might be, but obviously there are things that can get in there that I don't know how much it affects the small molecule side, for instance, maybe hair or even other things. On the large molecule side, that becomes incredibly that's a non-stopper on our side. That can't be in there.
I never, I never even thought about that, to be quite honest. So it's like, I can see why. Yeah,
Yeah. And then I should add, so we're in the middle of doing a study. It's an Australian clinical site. They're sending the sample here for us to do biomarkers on urine, um, biomarkers in urine, and a stabilizer is often used. And you talked about that, John. It just requires, not in our case, they sell urine stabilizers. So we're evaluating a couple off-the-shelf urine stabilizers for the impact on the particular biomarker that we're looking at. Interesting stuff, really cool things, and solving problems for people who, and in this case, it was a marker that you can do by like a chemistry analyzer, but they wanted increased sensitivity by using it. In this case, it's a colorimetric assay. So yeah, interesting, and really urine is something that we're seeing more and more samples of, and we've got a lot. I know we've got a long history of doing urine, especially in a preclinical setting.
I, I think it's easy to underestimate urine if you've never worked with it before, and so you expect it may be a simpler matrix to deal with, but it really is. It's always a challenge, and like I say, having worked with it, then you take a slightly different approach with urine compared to plasma based matrices, and I think experience is key with urine assays.
Agreed. I mean, I think if anybody's listening, we touched on 4 or 5 issues with urine, and so let's we can go on, and maybe John, you want to just touch on ocular real fast, and then we can dive into tissues.
Yeah, so ocular is interesting because it's a mix of fluids and tissues. Usually, with animals, you're talking rabbit or I suppose rat tissues. There's one assay that where we were working with where actually rabbit tears was the matrix of interest, and so the challenge there was actually dealing with a sample size of just a few microliters and trying to develop an assay based on that.
Yeah, definitely. And we're in the process of doing something similar. We have I've only worked in rabbit ocular. I'm not sure of any other preclinical models there. And then we have done some human tear studies as well, which makes my eye twitch even thinking about that. So in the rabbit setting that we're looking at, the couple things you can do, you can use like a half well, like so they can you use a well that's got a lower. So the standard well has a volume of say 200 microliters, and you have some that can go down to 100, and it just makes it lower. But what we really want is a more sensitive platform. That tends to be a way to resolve it. So meaning if you just take that 1 or 2 microliters and you QS up to 50, you can get some detection level on it. And then there are some, platforms that are specifically designed, 3 and 4 well plates. Our SMCX Pro is designed to look at things like ocular and
Sure.
fluids.
Yeah. Yeah. Yeah, I mean, it's quite a niche in terms of bioanalytical, but it is interesting when you get ocular studies to work on. I mean, I remember when we had our first dealing with tears, and basically samples come on little strips to then sample from that to then go further in the assay to mimic your plasma assay. It's challenging.
Mm-hmm. Yeah, we have some tissues that we're working on as well. Again, scraping the ocular eye or things in different segments of the eye. And I'm not an eye expert by any stretch. I know it's an immune privilege. I could go into deep detail around the immunology of the eye, but and then I also in the PK focus group that I sit on for AAPS. The eye has a lot of pumps inside of it. I didn't realize the rate of fluid going through your eye is very high from the aqueous to the humoral section of it. So in PK studies, I do know that you have different target ranges based off of which fluid you have, which I think is fascinating, right? So again, these are the types of things you kind of have to know when you take on these types of projects, right, John?
Sure. Yep, and another matrix we actually haven't touched on at all. I'm still holding off on tissue. Is feces, which again, it seems to be something we're increasingly seeing. I mean, having been in the industry for 20 odd years, I think the first fecal assay I was asked, or I remember being approached about, was only a few years ago. But it seems like it's becoming more and more prevalent, and it's got the unpleasant factor, and it's analytically challenging as well because it's sort of that halfway house between liquid and solid tissue. And yes, I'm really glad I'm not in the wet lab anymore because the thought of dealing with fecal samples just would horrify me. I mean, I'm not good with that stuff.
Well, you put in a fumer, and then, by the way, sometimes they are liquid samples, John, right? And then you can always put a little Vicks vapor rub under your nose. That's always a good trick with fecals. But for us, we're not seeing, there's never been a lot of fecal on a large molecule stuff. Thankfully, there's not a lot of studies. Now, there are some where people are looking more at like the microbiome, so it's popping up a little bit. That might be what you're seeing. But on our side, I never really, you know how many cells there are in fecal matter? Way more than you never imagine. You can fight call. I don't know if you know what fight calling is, but that's when you isolate peripheral blood mononuclear cells from whole blood. You can fight call fecals and find a whole bunch of cells in there. So, I hope that's not something we do. And that was something that I got to participate in graduate school. And by the way, that was probably the most unpleasant thing I did in graduate school was having to participate in fight calling of fecals. So,
Actually, to be quite honest, just just changing my children's diapers was something I avoided enough because it was so unpleasant. So I thought of doing it in the lab setting. I'm really not up for. I, let's move on to what probably is the most challenging of all is tissues. I admittedly have very little experience, though in my PhD did a fascinating project which was just like an academic exercise, but we my PhD was on SFCMS, supercritical fluid chromatography, and upstream of that we actually had supercritical fluid extraction. So what we did was spike some drugs into some meat matrix. I think it was liver and kidney. Freeze dried it, then extracted with SFE into the SFC and into the mass spec, which was a fun thing to do, but never seen a practical application of it since. So moving on to tissues in the KCS environment.
Okay.
Um,
I don't, John, you used a whole bunch of stuff that went right over my head. I hope somebody out there understood that. But no, on our side, really, you were. I think you were going to touch on some of the challenges. I'm going to take a small step back. Really, they kind of come in two flavors. We think of them as hard tissues and soft
yep.
tissues. What I mean by that is things like brain and even like heart. Heart falls into soft. Maybe even some of the standard like things like your quadriceps and the solus and other things like that. And then you get into harder tissues which and liver is soft tissue. Harder tissues are things like the bladder. Again, I kind of put the eye in that category as a hard tissue. So there's some. What am I missing on hard?
You got nails is another one that
Oh
we've
yeah,
been asked to do. I mean, so basically, what the challenge for tissues is, you have a solid, and obviously some tissues are more like softer than others. But really, we want that to be liquid, so we can then aliquot to then go for the next steps down into in our bioanalytical assay. I think we had challenges in preparation of standards and QCs. It does always a debate about, if you're just spiking a solution into the tissue, how can you guarantee that's representative of how your drug is behaving in the tissue? I'm not going to touch on that now, but that is definitely a question that we have. And then the next step is, how do you get what's the best approach for getting that tissue into a liquid form for sampling?
John, you're absolutely right. The tissue, the way we process the tissue to get it into a liquid format is through beads. And the instrument we like is called the Procellus. It's an automated instrument, and how that works is actually it works almost like a paint shaker, right? They put these small Cephedex beads in there, and then it literally shakes it like a paint can will. And in grad school, by the way, we used to use a paint mixer. We had one in there, and we put steel ball bearings in there, which is not a good GLP environment. These are much more controlled. The beads that are in there come in different sizes and shapes, and it's a nice
Yep.
soft way to mix it up rather than some of what we used to do in grad school. And then
Yep.
that gets pulverized, and then you also sometimes you use a little bit of detergent in there, and it works very nicely on most tissues. And we have I think to date, we have about 4 different protocols that we can use and help clients with in terms of what tissues and, optimal homogenization to allow for PK and biomarker analysis.
Yeah, the nice thing about the Procellus as a tool is, obviously depending on the size of the tubes, you can run tens of samples in parallel. So for a lab like us, where we're trying to obviously do as many samples per day as possible, that's a good tool to use. I mean, most in my sort of postgrad days when I was doing tissue work, which wasn't very much, we were homogenizing with a single probe. And the concern there was, yes, you'd break up the tissue, etc. You make the liquid, but then it was doing it one at a time. It made horrendous, not whiny noise. And then there was the big issue of how to prevent carryover from sample to sample. So the
Yeah.
Procellus option, where you're working in parallel, takes a lot of that risk out of the way.
Yeah, you're talking about sonication is what you were doing there, and that is definitely, it's a good way to do it, but it's not the reproducibility is low. And now I think we can do 24 samples at a time, and we've got 3 or 4 of these Procelluses that we can use. And it's like you said, they come in all different shapes and sizes. They're the Procellus machine itself has about 5 or 6 different. So from low volume down to like half a mil type size to 10 mil tubes, and now they sell the beads already in there, and it's just a matter of popping in, weighing it without the tissue, dropping the tissue in, reweighing it, homogenizing it, and then a very important step after you homogenize it is to get a total protein assay. That's the BCA test, and then you can start actually doing the analysis, whether it's PK or biomarker.
So I've never done the BCA total protein assay. I know you've talked about it in the past. So what does that actually involve?
It's a colorimetric assay. It's just you use an albumin standard for it, and so basically what it tells you is through copper binding, there's a dye that will get in there, and the assay takes a couple hours, and you put it onto a spec reader, and it tells you the absorbance, and you read it off the curve. I hope that all made sense, John. So it's just
Yep.
looking at
Yep.
I believe it's a copper binding reagent that is used to look at, and again, you're quantifying it off of an albumin standard. It
And,
works
and
nice.
then and then I assume that's used to normalize things. Okay. Okay.
Yes. So, for instance, if you're putting 50 microliters in of the homogenized tissue, the first thing you need to know is, do I have 1,000 or 10,000 off of my total protein measurement? And so then whatever your data bit is that you're reading out, you use that as a ratio of the BCA. So you get two numbers to come together to form a third number. Make sense? That's
Yep.
typically, there's a lot of ways to do that. I mean, I'm simplifying it, but the math behind it can sometimes be challenging as well, because sometimes you're doing multiple analytes or you're trying to do a couple different things that can create even more um, uh, um, again, calculations that can be tricky.
Sure. Yep. I mean, I suppose it's a similar thing with, but going back to urine samples, I'm really not as obsessed with urine as I seem to be. But obviously, you get different volumes from sampling from the patient, etc., and so you'll also do a normalizing with a creatinine assay. So it's some similar approach where you obviously have to you have to correct for you're using other measurements to help correct your analytical data.
Yeah, no, and we have a ready to. No, that's John, a really good point. We actually have a creatinine assay, not creatine, creatinine assay up and running for urine samples as well. So along with the BCA for tissues, we have the creatinine and we do that by colorimetric assay as well, or an ELISA-based method. So that's good, John. You say you're not obsessed with urine, but you might be one of those guys that thinks they got if you get stung by a jellyfish, that's myth. That's not going to work, right? That does not do anything to urinate on one's self after getting stung by a jellyfish. That's a myth.
Talking as someone whose father was a marine biologist, I'm well versed in that kind of stuff.
Well, John, that's going to wrap up podcast number 16. We're going to move on to our feedback. I'm not sure if hopefully we've got some. But while we're waiting for that to come up, John, what's some exciting stuff going on in your world this weekend?
This week, not a lot exciting. We are moving forward with the redesign of our backyard, so that we had the company in to talk to us yesterday. So hopefully that will get going in the next couple of weeks. I think that's largely it.
Yeah.
Yeah. Oh yeah. Actually, the other thing is, I mean, the contrast in my life from year to year. So today is August 13th. A year ago today was when my son and I set out from Boston for our first trip ever south of the equator, and 2 weeks in Madagascar. So we've been sort of getting very nostalgic over that and thinking, how on earth are we going to ever get to do that again? But yes, that's probably the biggest thing.
Yeah, and we were really I have such an uneventful weekend planned, but that's okay. Sometimes that's all right. But it seems to be the routine here in COVID 2020 summertime. But I don't, at this point, I'm just looking forward to um, you know, maybe just not working, right? There's nothing wrong with having a weekend where you're not working. Oh, and of course, it's tomato season, so I will be making pasta sauce. And my last weekend, I went to Nebraska to visit some family, and I brought back a full case of tomatoes. So I'll be cooking all weekend long, and with some luck, John, if you're ever here, I'll give you a jar of that, and you'll love it. It's one of my favorite things to do. I'll probably make a good, I don't know, 50 or 60 quarts of pasta sauce. I have so many tomatoes, and that'll be a couple days worth of just getting it on the stove and then canning it.
So,
I
so
would
what's
say like 12
what's what's the basic recipe?
hours. Well, John, this is a long passed down recipe, but I'll share some of it with you. So it's basically you cut the tomatoes and in the blender, you mix it with some garlic and basil. And for me, I use about 8 full of the blender, and then let that stew for a while. And you use tomato paste. So I use about one small can of tomato paste per blender, and so I need 8 of those. Again, usually I let the first hour kind of set in and get all that paste going, and it starts to bubble. Then there's sugar, Worcestershire sauce, Italian seasoning, some oregano. Remember, I already got the basil in there, that fresh basil,
Sure.
fresh.
Yep.
And then marjoram. I don't know if you know what that is. That's a cool
Yep.
Italian seasoning. I'm sure you do. You know a lot more about this than I do. And then, like I said, the Worcestershire is probably my secret ingredient that goes in there. And then I make a couple different. Sometimes I spice it up with some heat. I'll put some crushed red pepper, or sometimes I might even put in some garlic sriracha paste. That I
That
like.
sounds it sounds pretty good. I am a big advocate of Worcester sauce. So just a bit very quick bit of background. My father's family from Herefordshire, and the county just north of it is Worcestershire. And a while back there was a sort of rezoning of counties, so Hereford and Worcestershire got merged into Hereford and Worcester, and both counties hate each other. So it wasn't a particularly successful experiment. They've now broken back out to the two separate counties, but Worcester sauce was something that I was introduced to from a very early age. And my tip for Worcester sauce is when you do fried eggs, do a couple of splashes of Worcester sauce on the cooked egg, and it's top notch.
Mm. Yeah, interesting. And even like when you talk about towns merging and then splitting, I don't think anything like that happens in the U.S. It's so weird when you talk
Yeah,
that way.
county rather than town, but yeah,
Well,
it was
either
it
way, the
was
towns aren't merging, not since like colonial times when it was the Massachusetts Bay Colony back in like 1600s.
Yeah, it's in the mid 70s there was a bunch of rezoning of counties, so some disappeared and got swallowed into bigger counties. But then more recently things have changed back, but not for all those changes. Yeah,
You know,
I don't know what was behind it. It was way before my time. I was, but anyway,
I guess
yes,
even as I sit here, I take that back. Middlesex County, where I grew up, was redistributed in the early 80s. So maybe I'm just not thinking about it. But anyhow, John, that about does it for this week.
yep.
Uh, Exciting 16th podcast. Next week, our 17th, which is really cool that they've allowed us to do this long. We're going to go ahead and look at the differences and similarities of small and large molecule bioanalysis with a focus on PK. What do you think about all that, John? I think that'll be pretty cool.
Yeah, absolutely. I'll look forward to it. I'm sure we can find plenty to talk about. It's not
Yeah,
exactly a
no.
challenge.
Well, I think the challenges come around, and we'll talk more about it next week. But it's really like what regulatory requirement, and then what level of investment do you need? So that's what I
Sure.
mean by the challenges there. So interesting stuff. Well, that'll do it for this week. Any last words, John?
Nope. Thank you very much for the time, and look forward to doing this next week.